Hey guys, welcome to day 2 of cellular transport. And today we're actually going to look at how cellular transport is able to maintain homeostasis at the cellular level. Remember, last time we talked about the fact that there are two different types of cellular transport, and those two types are active transport and passive transport. The difference between the two is the way the particles actually move across the membrane. Passive transport, particles are going to move with the flow. It goes with what we call the concentration gradient from an area of high concentration to an area of low concentration. And because the particles are going with the flow, with the natural order of things, it does not have an ATP requirement. That cell is not required to use any of its own energy. Active transport is the opposite of passive transport. In active transport, particles are actually being forced to move against the natural flow. So they're being forced from an area of low concentration into an area where it's already highly concentrated. And the way that that is done is by using some of the cell's energy to make that type of transport happen. So let's talk about what concentration and concentration gradient actually means. When we say the word concentration, it simply means like the number of particles in an area, in a specific area. And if you add in a semi-permeable membrane to separate two regions from each other, anytime there is a difference in the number of particles across that membrane in those two different regions, then we have what's known as a concentration gradient. And that's what we have happening in cells. There's always a concentration gradient because there will be different numbers of particles on the inside of the cell than you have on the outside of the cell, or even within the different organelles that are in the cell. Sometimes you'll have a concentration gradient across the membrane of organelles, like we see with the ER and calcium ions. So a concentration gradient is simply the difference in the number of particles across a membrane. Now, the goal is for particles to move until they reach what's known as dynamic equilibrium. And so here we have a demonstration of some balls, and as you can notice, the balls were in one area was highly concentrated, and once they started to move, they started to bump into each other. Those random movements is what actually drives passive transport. So it's going to be those random movements that passive transport relies on, because as these balls or these particles move around, they bump into each other, they bump into their boundaries, and that causes them to change direction. And so that spreading out to an area of lower concentration is what actually drives passive transport. Um, Now, once a particle reaches what's known as dynamic equilibrium, once the particles reach dynamic equilibrium, they do not stop moving. Dynamic equilibrium is what happens when you have the same rate and amount of movement from one side of the membrane to the other side of the membrane. The example I use is like a bank account. If you had $0 in your bank account, and your parents deposited $100, then you had $100 to go into your bank account. But if that same day you spent $100, then you would have $100 come out of your bank account. Now, the net amount of money in the account will still be $0. So there's no net change in the balance of your account. However, there was movement that happened. $100 went in, and at the same time, $100 came out. So that's what we mean by dynamic equilibrium. It's not that there's no movement. The particles don't reach equilibrium and just stop moving. It's just that once they reach dynamic equilibrium, you have the same rate of particles going in and out of that cell or across the membrane in either direction. So when we look at these three diagrams here, we see that we have a concentration gradient. Two of them show the concentration gradient where there's a significant difference, and one shows dynamic equilibrium. So, which one of the three do you think shows dynamic equilibrium, where you have the same rate and amount of particles moving in both directions across the membrane? All right, hopefully you chose C, because C is where we see the dynamic equilibrium, where particles are going both ways across the membrane. They're no longer moving from a higher concentration to a low concentration only. And this is what we mean by dynamic equilibrium. So, to passively go from high to low, the cell will use DFO. D represents diffusion, F is for facilitated diffusion, and O is osmosis. Now, diffusion is often called simple diffusion, and this is what happens when particles are able to actually slip through the spaces between the phospholipids and be dissolved across that internal nonpolar region of the phospholipid bilayer. We talked about this when we were talking about the structure of the phospholipid bilayer. And remember, the particles that are able to do this first have to be small enough to slip through the spaces, and then they also have to be nonpolar to be dissolved across the interior of the membrane. So these particles are going to be able to move from a high concentration to a low concentration, and without the use of energy, but they have to be both small and nonpolar. And substances that can do this can do it quickly. Two great examples of particles that do this would be carbon dioxide and oxygen. And that's because we need to be able to get carbon dioxide out of our systems quickly. It's a waste product, and we need to be able to get oxygen in. And up here, there should be a capital O for carbon dioxide, not cobalt. I don't know how that happened. Sorry. All right. So now, remember to passively go from high to low. The cell will use DFO. So we've talked about the D, which represents simple diffusion. Now let's talk about the F, which is facilitated diffusion. Now, remember that the word facilitate means to help. So what's happening with facilitated diffusion, particles are still moving from an area of high concentration to an area of low concentration, but they need help to get across the membrane. And in this case, their helper comes in the form of a protein. This is an example of a protein channel because it's open. But remember, transport proteins can be either channels, or they can be gated, or they can also be carrier proteins. But the point is that anything that cannot slip through the cracks and be dissolved across the membrane has to have another way to get in or out of a cell. And they do that by going through a transport protein. Now, transport proteins will allow things that are large or hydrophilic, or as we say, large and charged. And remember, the two types of charged substances would be our ions or our polar substances. So anything that's large or charged would have to go through a transport protein from an area of high concentration to low concentration in passive transport. An example of that would be like glucose molecules, which C6H12O6. So glucose is kind of large, so it won't be able to actually fit between the spaces of the phospholipids. And then you have things like sodium ions or potassium ions, where even though they are small because it's only one atom, they carry a charge. And so even if they could fit between the spaces of the heads, because this area is hydrophobic and these ions are hydrophilic. Remember, like dissolves like. They would not be able to be dissolved across the membrane. So instead, they have to bypass the interior of the membrane and go through a transport protein. But again, because they're moving from an area of high concentration to an area of low concentration, they go with the natural flow and they do not require the use of ATP. So again, to passively go from high to low, the cell will use DFO, diffusion, facilitated diffusion, and finally osmosis. Now, osmosis is the facilitated diffusion of water only. Osmosis talks about how water quickly moves across a semi-permeable membrane from an area of high water concentration to an area of low water concentration. It is very important that you remember volume is not the same thing as concentration. As we see here in the diagram below, right now we have equal volumes of water. That is the equal amounts of water on both sides of this membrane. However, we do not have an equal concentration. So when we're talking about the concentration, we're talking about in a solution where you have water as a solvent and you have other particles as the solute, what is the amount of water that is available per solute molecule? That's our concentration. So here, what we see is there's more water available for this amount of solute molecule than over here. If you were to divide each one of these little dots that represents a water molecule, and then you divide it by the number of solute molecules you have, which in this case is sugar, we see that there's more water available per solute molecule on the left side than on the right side. And water moves from an area of high water concentration to an area of low water concentration. So because of that, as the water moves, it's going to move from the left side to the right side to balance out the concentrations of the liquid with the water with the solutes. Now notice what happens to the volume though. By doing that, by making the concentrations equal, we made the volumes unequal. And that typically happens. That's going to happen sometimes. So remember, we are not looking for equal volumes. We're looking for equal concentration. Now, osmosis is only talking about how the water moves, not how the sugar moves, not how another particle moves, but only how the water is going to move. And water moves through special proteins called aquaporins or water pores. So water movement is a special type of facilitated diffusion. It just has its own name. So it's moving from an area of high concentration to an area of low concentration without the use of ATP. And because it's the movement of water, it is called osmosis. And that movement of water is able to create two types of pressure. The first type of pressure is osmotic pressure or water pressure. And that pressure is going to is going to be created in all types of cells. The second type of pressure is called turgor pressure, and we see turgor pressure in cells that have a cell wall, like a plant cell. Because of that, water actually affects animals and plants differently because of plants' ability to have turgor pressure. So, which two of the following are examples of passive transport? Remember, to passively go from high to low, the cell will use DFO. So when we look here, we see that we have osmosis and we also have diffusion. So let's take a closer look at osmosis in the next video.